Even when I was a little kid, I was really interested in things like zoology, but then that developed into more appreciation of the breakthroughs associated with molecular genetics. I have done a lot of basic research, first as a graduate student, and then here as independent faculty, I was able to explore really any avenue. And so I got away from some of these basic questions into this more translational work, which is exciting because we all have good ideas. At least we like many of our ideas, and we want to see those become something that helps out society. My lab studies many aspects of RNA biology, but one of the things that we've done is try to apply our insights to develop a technology. In particular, what our goal is to use different RNA designs to target pest species. And what this does is it allows a completely biocompatible molecule that's very environmentally consumer friendly so that you can eliminate a pest without any of the drawbacks of toxic pesticides or other things that might be bad for consumers or the environment. Resistance is a perennial problem. All types of pesticides, all technologies, the pests develop resistance. Even the RNAs that we develop, we know that the pests are able to develop resistance as well. Now, one of the benefits is that it's pretty easy to recode them. And so we should be able to quickly address pesticide resistance versus develop entirely novel molecules for those purposes. RNA is in every cell of every life form. It's a common feature of cells, and it's how they convey information within themselves. And if we can develop ways to interfere with that, then we can control the cell. RNA interference is a technical term, actually, was established decades ago as a method to do gene silencing. It exploits a particular way cells process RNAs and then recognize things to be silenced. By using RNA interference, we should be able to introduce a sequence that targets an essential gene within that pest, which would kill it. Now, the useful thing about this is that the genes in the pest are very different from the crop, the humans that might consume it, or even other insects in the field. And so those will not be affected by that very specific sequence. And so it's precision removal of an animal from the crop plot. You would leave all the other beneficial insects like bees, butterflies, unaffected. Where current technologies rely on these sort of broad spectrum insecticides or transgenic plants that, again, they are anti-pest, but they're also anti any insect. There are actually currently multiple products on the market that use these sort of approaches, whether they're transgenic horns or even sprays where you can spray an entire field with an RNA molecule and target specifically animals of interest. My lab comes in where we develop additional evolution of these approaches, exploiting novel mechanisms that might be better for certain pests that are not really addressable by the state of technology out there right now. The Mississippi Smart Business Accelerator Initiative was extremely helpful for our research. It allowed us to explore some new ideas, create some preliminary data, which then led to a larger grant through the National Science Foundation that will really enhance translational research at the University of Southern Mississippi to create new jobs and build up industry for the community. So we received some funding through the Mississippi Smart Business Accelerator Initiative, which allowed us to apply for an NSF ART grant, which is Accelerating Research Translation Grant from the National Science Foundation. This is a large multi-million-dollar grant to Southern Miss. Part of that is an association with a mentor institute. And in this case, it's the University of Arizona. Their technology development office can work with ours to become a more translationally oriented university. And so that collaboration led to the interaction where the two institutes began to work together, and that led then to the submission of the SMART Act, which we got. We were one of hundreds of different institutes in the country that applied. So it was very encouraging to see the excitement that the National Science Foundation had for the work at Southern Miss. As the project goes forward, we're also including additional participants such as my colleague Tristan Clemens in Polymer, and they're going to begin to use their expertise to make our product even better.